up in the podium.
We're probably going to have an adventure here because I have a stack of questions about like this. So our dear panelists who are world-renowned scientists are going to be playing the game of the lightning round of questions, I have a feeling. But let me do a quick introduction because three of these scientists were in the film you just saw. Dr. Walter Bradley is a world-renowned expert in neurology, neuromuscular diseases, and ALS, and was awarded the prestigious Forbes Norris Award of the International Alliance of ALS-MND Associations in 2006. He's a professor and chairman emeritus at the Miller School of Medicine in University of Miami. Since 2007, he's been involved in research in the causes and treatment of ALS, teaching and writing books, and chairs the data monitoring committees for the pharmaceutical industry and international studies of developing new drugs for the treatment of neuromuscular diseases. Dr. James Metcalf, yes. Next to him, Dr. James Metcalf began working with Paul Cox at the Institute for Ethnomedicine in January 2013. He's a senior researcher studying cyanotoxins. Dr. Metcalf was educated in the UK, in Wales and Scotland, and holds a Bachelor's of Science in Marine Biology and Biochemistry, and his PhD is focused on immunodetection of cyanobacterial toxins. He's continued to examine cyanobacterial toxins throughout Europe, and his current research includes the particular neurotoxin amino acid BMAA. Thank you for being here. Dr. Larry Brand is a professor of marine biology, and your team is here apparently, and ecology at the Rosenstiel School of Marine Atmospheric Sciences at the University of Miami. He's investigated harmful algal blooms for many years throughout the greater Everglades, including Lake Okeechobee and the Calusa and Indian River Lagoon, St. Lucie Canal, and many other Everglades water bodies. He's studied red tide also along the Gulf Coast of Florida. His past work has focused on toxic algae in the water and in seafood. He now is initiating research on algal toxins that get into the air and the human health concerns that aerosolized toxins present. I first met Larry about 10 years ago when he first put this issue of BMAA and toxins with cyanobacteria on my radar screen. John Cassani is the Calusa Waterkeeper. His professional career as an ecologist started in Lee County in 1978. He has degrees in biology and fish and wildlife. For 36 years, he worked as a resource manager for local government, managing waterways of Southwest Florida. He's a courtesy faculty member at FGCU, Department of Marine and Ecological Sciences, with research interests in areas of exotic species and biodiversity. So, here is our tremendous panel, ready to answer your many questions. So this is a challenge, and I'm going to ask these scientists to bear with me as we try to do maybe something like a lightning round. We might be going back and forth because I didn't have too much time to sort these questions. But we want to get to as many of them as possible because you've got tremendous questions that you've got here. What we know is that this film is applicable to our community because in June of this year, we started to see a massive cyanobacterial bloom coming out of Lake Okeechobee into our coastal waters, our river and coastal waters, as well as the East Coast. And this is probably one of, I would say undoubtedly, the worst bloom that I have seen, and I've been here 40 years. So I want to make sure that we touch on these issues with the cyanobacteria and the freshwater blooms and the toxins, and then expand our discussion out to the other problem we're having the red tide and the toxins and marine life impacts, health impacts of that as well. So I'm actually going to go right to the questions. How do we find out, and I'm going to pose them, and there's a free-for-all that's going to happen on the panel. How can we find out if BMAA is in our drinking water and if our drinking water is safe? Because as the film said, they can detect toxins and treat for toxins, but it also said there is nobody testing for or a way to test for BMAA.
Yeah, can you hear me okay? Well, yes, you can test for BMAA. There are many ways of doing it. As Rayanne mentioned, I work at the Brain Chemistry Labs in Wyoming. We use mass spectrometry, and we weigh the toxin molecule in cyanobacteria, in fish, and other samples that we have. We've never actually found it in water, but there are no commercial tests. We are working towards producing a commercial test similar to how you would test if you were pregnant by using antibodies. And that's hopefully coming up in the next few years, and we're just trying to get that going right now. But in terms of drinking water, we haven't seen it in purified water. But as we do more research and do more testing, that may change. But right now, it's probably more where you find cyanobacteria that you're more likely to find BMAA.
And would you be likely to find it in bottled water?
Depends where the bottled water comes from.
Well, there you have it.
I mean, certainly with cyanobacterial toxins, if you look at, say, for example, the issue of microcystins and primary liver cancer. So in the 90s, there was a number of studies saying that if you were exposed to low concentrations of microcystin in China or in Serbia, you had a higher incidence of primary liver cancer. The interesting thing from the studies in China was that they just compared people who drank from surface water ditches versus those who drank from well water. The ones who drank from surface water ditches are the ones that had the higher incidence of primary liver cancer. So it comes back to your point about bottled water. If it's from an aquifer, which may not be great for the environment, it's probably better for cyanobacterial toxins, whereas surface water with high nutrients, higher cyanobacteria are potentially more likely to contain those toxins. And as long as your treatment facilities aren't working very well too, because obviously bottled water needs to be treated. So again, you need to make sure that those treatment facilities are working well to remove any potential toxins that may be present.
But there's a distinction between the toxins and the BMAA. So the Sandusky plant was talking about removing the toxins from their finished water.
Correct.
But there wasn't a test?
Well, with the Sandusky, so Ohio has regulations for drinking water and for recreational water. Those guidelines are for toxins like microcystin. Maybe Slimnus, but generally when you hear people talking about toxin testing, they're talking about microcystins, which we know exist in Lake Okeechobee and in the Calusa and in the St. Lucie River. So that's what they're talking about when they talk about toxins. Mm-hmm. BMAA, there's not testing, because there's no one's going to test for BMAA because no one is required to test for BMAA. And that's ultimately what it comes down to. Is if you don't have to test for it, then why would you test for it?
So in our Olga water treatment plant upstream, where there is a water treatment plant that takes water out of the surface of the river, that can be treated for toxins, but it's not being tested for BMAA.
That's my, yes,
Yeah.
that's my understanding.
Okay, just want another one down. Do we know if BMAA has been isolated in any of our waters locally?
Yes.
Yes.
We have a paper coming out from the 2016 Bloom samples, and we did find very low amounts of BMAA in some samples collected. And one of the reasons why we are here is we're collecting samples again to continue to build in our data sets to try and understand long-term trends and what's going on.
But Rayanne, we should say that when you ask that question, is BMAA in our waters, not if you've got the bacteria out of it, but if in fact you go swimming in it, you're going to be exposed to a tremendous amount of BMAA.
Okay, so that leads to a question about the most risk. Is the most risk from BMAA from aerosolized inhaling of the toxins, from physical contact, swimming, swallowing, or from eating fish?
Yes.
Okay, there you have it. A definitive answer. I told you it would be a lightning round. Have there been an assessment of the number of ALS or neurodegenerative diseases along the Calusa-Hatchie or water bodies impacted by cyanos here in our local community?
So we have been able to analyze the distribution of ALS patients in about 1,500 such patients, and there are hotspots really scattered throughout Florida. Some of them are in areas where there are these blooms, and we're trying to now extend that research to be able to tie down the concentration of cyanobacteria and the frequency of ALS. So yes, there undoubtedly are hotspots here in Florida of ALS and other neurodegenerative diseases.
Are they local? Lee County, the coastal, the inland counties in this region?
There are some on this west coast of Florida, yes.
Do we know what they are? Can you say?
At this moment, we haven't got enough evidence to be able to answer that question.
Okay, okay. Were there any epidemiological studies done with the people who reported health problems on the other coast in the 2016 bloom?
No, you're talking about green blue algae on the east coast of Florida.
In Stewart and Central Marine, that area?
There are certainly, there have been the acute toxic effects that have been reported, and people have had problems, skin rashes and other things of that nature. This is research which is still ongoing, and this is one of the difficulties. We know that there are acute toxins. We know that BMAA is present in those cyanobacteria. We know, and you saw it in the film, that if you take pure BMAA and you give it to animals or tissue culture or anything like this, you can produce a very severe disease, including things that look like Alzheimer's, Parkinson's, and ALS. We have no protection that is offered to the public about these chronic illnesses. So this is one of the difficulties that is there. The research, in fact, needs to continue, and public protection is what is needed in terms of the advice that is being given to the populace.
Is there a test other than a brain sample to see if you have BMAA in your system?
No.
No. Good. So no surgery this afternoon. If sugarcane is being irrigated by Lake Okeechobee waters and they have known cyanobacteria blooms in them, could that be getting into the sugarcane like as in the movie it was getting into the wheat?
Well, it could be. On the other hand, what you're eating out of that is the sugar, so you wouldn't be getting much BMA in the sugar part of the sugar cane.
Because there's no
protein. Because it's supposed to go into the protein, yeah.
Okay. Is there an argument for growing cannabis instead of sugarcane? I just have to say, that was a pretty common question in this crowd. I'm not judging, just
that's for discussion outside this auditorium.
I should point out, there's an even better argument for growing sawgrass in the northern third Everglades, where you currently have sugarcane.
Native plants win out. Let's talk a little bit about this Elserine, the amino acid that is in this death fight with BMAA, two amino acids going at it in our cells. Is there a particular supplement that people might take to help prevent ALS, Alzheimer's, or cancer, or Parkinson's?
So let's just go back a fraction to the animal experiments that you saw depicted in the documentary here. Elserine definitely lowered the amount of damage to the brains of monkeys that were being treated and limited the amount of damage, therefore was either preventative or certainly a treatment if you happen to be a monkey. All right.
And aren't
We
all?
have done trials at this moment in a small number of ALS patients and a small number of Alzheimer's patients. And in the early trials of Elserine in ALS, it definitely seemed to produce a reduction in the rate of progression of the disease, particularly in those that were on the highest doses of Elserine. So that's where the, if you like, hard scientific evidence currently sits. There are a large number of people who are taking Elserine because they hear that it's just a supplement, it has really no toxic effects, and therefore they are taking it as well. And there is anecdotal evidence that a fair number of patients with these neurodegenerative diseases, as you saw Ellie in the documentary, did feel that it was in fact benefiting her very significantly. We are now in phase 2 trials with larger numbers of patients of both ALS and Alzheimer's, trying to see if we can produce definitive evidence that Elserine is effective. That's part of the story, and I think we can leave it there. But the part of the story which is relevant, perhaps, for why we are here today, is maybe we can prevent the disease is coming in the first instance, and that's another very important piece.
And just building on that, are there any tests that, plasma or blood tests or anything else that, having had this exposure in our community for the past 2.5 months, that anybody should be asking their doctors to run?
Sadly, no.
No. Okay. So, who's eating fish these days? Have you stopped eating fish? Are there any sources like farming sources that would be healthier than wild sources?
I've never been much of a fish eater, and I live in Wyoming, so it's really hard to get good fish in Wyoming. So certainly from like the work of Professor Brand, there are certain species that seem to accumulate BMAA. We've seen BMAA in blue crabs from Chesapeake Bay, which we published 5 years ago. Larry's work on, where was it? Down south, wasn't it? Florida Bay. That was it. Yeah.
River.
Has shown BMAA present to be in certain fish species. We need to do a lot more work to work out, is the BMAA there all the time? We don't know. Certainly when you eat shellfish, marine shellfish, you often depurate or leave the animals in sterile seawater for 48 hours. That allows them to clean themselves out and get rid of some of the harmful microalgae that they may be exposed to. So ultimately it comes down to testing, I think. If you can test your shellfish or your fish when you catch it and say, okay, right now we have a bloom of cyanobacteria, we have a bloom of Carenia, maybe it's a good idea to close down this particular fishery until the danger has passed. But that also then comes into guidelines. Without the information, it's very hard to make informed decisions. If someone says potentially that there might be toxins in your fish, we don't want to cause panic. That's not going to help the situation. You need to test the fish and then work out which fish are contaminated at which point in time, and then find ways to remove that contamination at that point in time, and then bring in the guidelines, because we also don't know how much BMAA is required to cause an effect in everyone, but it may be that with the case of ALS, just 2 in 100,000 people are susceptible because of their particular differences in genetics.
So stone crabs versus bivalves, equally blue crabs, stone crabs, bivalves.
It all depends on the scenario. I mean, it's very hard to say.
Yeah, we found really high concentrations in blue crabs, both in Chesapeake Bay and in Biscayne Bay. In fact, twice as high as what were found in those fruit bats from Guam. So
wow.
It seemed to be, we hadn't had that hypothesis, but just scanning through our data, it looked like it tended to be fish and animals that hung out on the bottom fish, and then the crabs and shrimp. We didn't test stone crabs, but the blue crabs had high levels, and it wouldn't surprise me if stone crabs would have high concentrations as well.
Are there treatment technologies for treating that are commercially available for treating the algae to eliminate it?
Yeah, but there's lots of ways in which we could kill algae, and I get companies and people give me new ideas all the time. Oh, and we've got a new type of snake oil that'll kill algae. The problem is these cyanobacteria are really tough. Almost anything you use to kill these algae and kill a lot of other things in your ecosystem. It's like you have to kill your ecosystem in order to save it or something. Mm-hmm. The other aspect of this, Lake Okeechobee at one point is like 90% covered with these algae, and it's like, what, over 700 square miles. Logistically, how are you going to add anything to get rid of those algae? Same thing, I think this red tide along the west coast is like over 1,000 square miles now. Mm-hmm. Logistically, how are you going to do that? The other aspect, okay, so you kill these algae, they die, they release all those nutrients back into the water, you stop adding this toxin, whatever you're doing, they're going to just go right back again. So it's like you're treating this natural ecosystem like a swimming pool. Sure, you can chlorinate that swimming pool, kill the algae, and kill, if you had fish in your swimming pool, you'd kill the fish in your swimming pool as well, right? You stop chlorinating that pool, what happens? The algae are going to just grow back.
And the scale of this is so extraordinary that the questions about filtering, it's just an industrial scale.
Right.
What do you have opinions about the current proposal or the plan that's being implemented by Lee County to suck some of the dead-end canals and the crusty surface accumulations out of the system, so they're removing the biomass, they're removing that physical, and at least making conditions maybe safer, more pleasant? Do you have opinions about that?
Yeah, what happens, these cyanobacteria, they produce little gas bubbles. They float to the surface, and then the wind blows them to the shoreline or to the ends of these dead-end canals. So you end up with these really high concentrations at the ends of these canals where people have their homes right around them. So in that case, it might very well make sense to suck up just that small area, or maybe even add a toxin to just that small area to provide some temporary relief to the homeowners around there. But large-scale treatment is just not going to work. Doesn't make it.
But then where they're taking it.
Excuse me?
Where they're taking
But
it.
yeah, then what do you do with
question.
the algae
Brian,
sucked up?
Can I ask Larry to talk about nutrients and what is producing these blooms, the inflow of nutrients?
Yeah, the way to get rid of these algal blooms, or at least reduce them, these technologies that people are talking about for killing them, it just doesn't make any sense to me, really, on large scale. You really need to stop the nutrients at their source before they get released into our public waters.
When you find yourself in a hole, quit digging.
Exactly. And clearly our current regulations are either not good enough or they're not being enforced well enough.
So let's talk a little bit about nutrients. I think this crowd understands that that's the common ancestor to both of the blooms that we've been experiencing. The nutrients have a role. Do we have a need for additional standards, thresholds, requirements?
Clearly, yes. Clearly they're not working. There's no doubt about that.
Is there a reason that our beaches were not closed when this first showed up?
Yeah, I'm really not, as a scientist, I don't really keep up with all the laws and everything, but my understanding, what I was told, is that in this state, they do not have a mechanism for actually closing the beaches. They're not allowed to keep people from going to the beaches. They can put out warning signs, but they don't absolutely close the beaches. Now, some states have laws which they will close the beaches and keep people from going to them. This state apparently does not have that type of law.
John, do you have any thoughts on how the rules handle public notification, really posting points of access, boat ramps, beaches?
I think there's an acknowledgement and recognition that perhaps posting was a little bit chaotic there for a while. We got to the point where the bloom was extraordinary. Actually wrote a document to quite a number of policymakers about developing some leadership on that particular issue. And there's like 22 or 3 other states that have very progressive response strategies to posting. They have what they call numeric thresholds. So when microcystin gets to 10 micrograms per liter, it triggers policy actions. And we really need that. We need that more here in Florida.
So the only standards, thresholds that we've seen are WHO standards for recreation and...
Well, there's been decades of research now, and EPA updated some of their recreational drinking water guidelines in 2012, and there's a draft document 2016. So every time a new guideline comes out, it looks like the risk thresholds keep getting lower and lower. It's our recognition that the toxicity is worse than we thought it was. So those are out there. Those are federal guidelines that are out there now. Unfortunately, cyanotoxins are still unregulated contaminants in this country, so that's kind of holding back and basically turning it over to the states, say, you develop a program to deal with this. And there's a lot of states that, Ohio, Oregon, some of the Northeast states have very progressive response strategies now. And I think it's really an opportunity for Florida to get there, to get out ahead of the curve on this issue. Things like global warming. We know that cyanobacteria can outcompete many of the other species of algae at relatively high temperatures. So there needs to be a recognition of that, not looking the other way on things like climate change. I look at it now as more of an opportunity to do some really interesting and advanced work on this particular topic, not just public health, but ecological effects as well. So I see it as an opportunity, and it's a huge challenge. The public health aspect of this is an enormous challenge. And I think we can get ahead of the curve on it, but it's going to take some commitment, some courage, and some funding to do that. So,
Are there is there any type of respirator that's effective for both cyanotoxins and the brevetoxin?
I think the problem of the toxins is that they're very small molecules, and they'll go through any filter that essentially you can put up. The actual organisms can be filtered out by industrial level filters.
Yes, I mean, yeah, so the organisms will not go through a filter. I was thinking about this earlier. For brevetoxins, for example, they're volatile, so I mean, the answer is we don't know, because no one's ever tried to test what filter would it work to remove cyanotoxins that you're breathing in. But if I was going to go out and do some sampling, I'd probably go for something that takes away things like paint fumes, volatile organics that can get blocked by the filter, because brevetoxin should do a similar thing. We know it can bind to certain things like carbon 18, which is something we use in the lab all the time.
So those are rather like the gas masks that you're talking about, carbon filters and so forth.
Yeah, I think
this is not the thing that you saw Paul Cox wearing.
No, I mean, I came down here to go out sampling with John, and I went to Ace Hardware, and I'm looking at all the respirators going like, which one do I pick? And I picked quite a nice one, actually, so I'm hoping that's going to be really good. But again, it comes down to research. We don't understand, well, not understand is the wrong word. We don't know what level of protection is needed to protect even the scientists working on it, and we're the ones that have the information. But we can go out and test them. And also with you people, from my experience in Stewart over the last month, people have come to me and said, I'm having problems breathing, I'm having problems with sore throat, I'm having problems with my nose and my eyes. I think at that point you should probably maybe need to go and move away from any blooms or things that you're at if you start to feel those sensations, those symptoms. And certainly we've been seeing medical physicians who are asking people, have you been exposed to cyanobacteria? Because you are also going to provide us with information about what the risks to public health are. And as John mentioned, it's a massive challenge to understand public health. Panicking is not going to be good, but having guidelines and information about whether you recreate at this lake or not, whether you can drink water from this lake or not, whether you can breathe air from this lake or not, requires policy, because if you don't have to test for it, no one's going to tell you what the results are.
John?
Yeah, I just for my own edification, and a lot of people here tonight, could you raise your hand if you've had a direct exposure to this cyanobacteria event? So when I say direct, you live on a canal, you've been in a boat, perhaps you've smelled it. I would consider that a direct. Oh my goodness. Yes, almost everyone.
Pretty good.
Well, really appreciate your coming and learning more and being courageous about it and maybe being a little angry about it. So I share those emotions, but this has almost been a silver lining to this event. You know, I have had people calling me from Jacksonville, from Orlando. I don't know these people. They just want to come here and help with it. So there's this widespread recognition that we are being so disproportionately impacted here in this community, on this side of the state, this particular year. So my hat's off to those who are bearing with it, that have smelled it in their house, that couldn't get away from it or had to leave their home for days at a time, maybe missed work as a result. So the implications of this event are extraordinary. Economic, public health, it's just, I'm just blown away by it. And I haven't been to an environmental perhaps event like this where I saw more than 40 or 50 people in the 40 years that I've been doing it. And this is just incredible. So thank you for being involved.
So we're starting to see the algae wane, whether that's actually happening or not. There's a question here about how stable the toxins are. Once the bloom has gone away, you don't see it in the water column on the surface. How long are those toxins still present in sand, maybe in any kind of DNA for different species?
So the organisms first.
Yeah. Okay. And some of these things I'm not really sure, but I'll just give you one example. We're finding dolphins out in the Gulf of Mexico that are dying. There's no red tide around. Okay, but you do the autopsy, they're packed full of brevetoxin. What's happening is, well, I think probably a lot of you probably have this sort of guideline. If you see a dead fish out there during a red tide, you know not to eat it. It's exposed to brevetoxin. Some people assume if the fish is still alive, it must be okay. Well, turns out we now know a lot of those fish have what we call sublethal concentrations. Is that concentrations that it doesn't kill the fish, it's not enough of the algae that you can see it, you can see discoloration in the water, our satellites don't see it, it's low enough that you can't detect it and it can't kill anything, but it's still there and it's accumulating, this brevetoxin is accumulating in that fish tissue. Now what's happening over time, and we've documented this as well, I mean, even when there's no red tide, you sample all these fish out there in the Gulf, they've got brevetoxin in them. These dolphins out there now eating these live fish that have these sublethal concentrations of brevetoxin, over time they're accumulating more and more of this brevetoxin. It's just like methylmercury. You don't want to eat too many of certain types of fish because of the methylmercury in them. Okay? Over time, you keep eating more and more of this, it ends up killing the dolphin. Okay? So in this case, this toxin here, even though you don't see a red tide, that toxin, the brevetoxin, is persisted in that food chain for very long periods of time.
So there's a question here about whether lupus or rheumatoid arthritis might have any connection to blue-green algae.
If you swim in the green scum, you can get dermatitis and a variety of other skin diseases, but there is no evidence that this is related to lupus or rheumatoid arthritis, no.
Okay. And are there any testing of crops that we might know are contaminated with BMAA? Is there any kind of FDA testing? Obviously not for the spirulina health drink. Suspiciously looks like it, but who'd have thought that wheat...
The short answer is no.
no, okay.
But I mean,
No, they're not contaminated, or no, we don't know?
we don't know. I mean, we have, there's a few anecdotal reports from in the past of Many years ago, we studied some lettuce crops in the UK. And what happened was the farmer was growing lettuces, and he had a brick pond which he sprayed water from over his lettuces. And that pond developed a bloom of cyanobacteria, and those lettuces were too toxic to be sold to the public, and they were destroyed. So it can happen. We know that BMAA is quite metabolically active. It will react with things. Organisms will take it up, use it for various sources, and you can reappear as well. We've seen in certain shellfish that you can make BMAA disappear into the shellfish and then reappear when conditions change. So we don't test for it, and we don't know about it, but it is feasible.
So, John, we have a problem in the Calusa hatchie that we've nicknamed the Goldilocks condition, where we'll have these cyano blooms not just when we have high flows like we're having now, but also we have them in the dry season when we don't have a high flow coming out of the river. Can you explain what's going on there?
Yeah, I think that there isn't necessarily a direct link to flow. And what I do know about how we forecast, or the data being used to forecast cyano blooms, pretty complex. It's pretty complex models. And some of the thinking now is that ferrous iron in the sediments in combination with very low dissolved oxygen can be a trigger for these blooms. There's so much new information coming out that may be independent of season or regulatory releases, those kinds of things. It's tempting to say that one particular factor caused it, but it's really a complex suite of factors that trigger these blooms. At least that's what we're learning now. I'm really hoping that we'll get the Florida Harmful Algal Bloom Task Force back going again here. So we can use some of this breaking new information and get that out there in a way so we can manage these blooms, maybe even prevent some of these blooms. There's too much nutrients almost all the time, but we don't have cyano blooms all the time. So there's other things triggering these blooms. So I hope we can get ahead of the curve on that in the future.
So the other problem that we're having involves red tide in the coastal waters, in the marine waters. Is there a connection between the blue-green algae and contributing to an acceleration or a connection to red tide?
Well, the fact that you've got these blooms in Lake Okeechobee and on the Calusa River tells you there's a lot of nutrients in that water. That's now flowing into the Calusa estuary and now out into the Gulf. So there's a lot of nutrients flowing down there. On top of that, these algae, when they make it down there, these are freshwater algae, they eventually die in the salt water. That releases yet more nutrients. Those nutrients then get released to the red tide. So once you've got the red tide there, now with all these nutrients flowing in, you're just feeding. Excuse me. I did a study.
You got a little red tide there. Look, take a sip.
I did a study.
Take a sip of that good water in front of you.
Depends on where it came from. I did a study, and it's very clear. I looked at 50 years worth of data of the red tide here in Florida, and clearly there's a higher abundance of the red tide right there along the shoreline compared to offshore. It's a very clear gradient, much, much higher concentrations inshore where you're getting your land runoff.
So take us through. The red tides generally start 10 to 40 miles offshore in the bottom of the Gulf?
Well, that's one of the dominant hypotheses. I'm not convinced that's always the case, but it's okay if you want to argue for that. But it's got low abundance offshore. Mm-hmm. It seems that it becomes really abundant once you get inshore where you're getting the land runoff. The red tide itself is a natural thing. We know it's been around for at least 500 years, okay? But I also, in this study, I also found that it's like on average 15 times more abundant today than it was 50 years ago.
Okay.
And I can't think of any natural nutrient sources that have increased 15-fold in the last 50 years. Now, what obviously has increased dramatically over the last 50 years is us. There's a lot more people in South Florida than there used to be, and along with associated activities, agribusiness, lawn fertilizer, sewage, septic tanks, etc. And I think that's what accounts for this much higher concentration of red tide right along the coastline where your land runoff is occurring.
So in the news, you might see some of the agencies describing the red tide bloom as a natural occurrence. But Sanibel has just removed 270 tons of marine life at the cost of $74,000 a day from our beaches, and that is by no means natural. So after a red tide bloom is finished, how does one figure out if the water's safe for swimming, fishing, and what are the requirements for notifying the public? So we have a lot of visitors that come from all over the world. They may not be watching the news. They may not even speak English. So how do they know when they've come, if the red tide is done, that the water is safe, the beach is safe?
This may sound a little strange, but in a sense, I regard the brevetoxin as sort of a good toxin in that when you're exposed to it, you know right away. It's like a tear gas. You immediately have watering eyes, you start sneezing, coughing, etc. That's what's really bad about BMA. You have no idea you're being exposed to it until 10 or 20 years later you might develop these neurodegenerative diseases. So in the case of the brevetoxin, the red tide, if you're not detecting it, it's probably okay to go swimming. However, it's not necessarily okay to eat the fish just because, as I mentioned, people have shown that even if there's no red tide around, those fish still have, a lot of those fish still have, if it was a sublethal dose, they've still got some amount of brevetoxin in them. And what time span it takes before they eventually get rid of it, we don't really know. I mean, people have sampled fish that had not been exposed to a red tide for at least 6 months, and yet you still found the brevetoxin in them. And of course, again, it can biomagnify up the food chain, as I mentioned earlier, with respect to the dolphins, until eventually, it can be years later, the dolphins actually die from this accumulation of the brevetoxin.
So do we have much of a sense of that aerosolized toxin and how far away it can travel, how much breathing it in is going to start to have either immediate or longer term effects of either the red tide or the BMAA?
So aerosolization is a relatively new phenomenon with regard to these toxins, and the evidence is still being accumulated. Legionnaire's disease is an aerosolized distribution of the bacterium from water towers and other things of that nature. And there is more evidence in that field, and the distribution from the sources of those bacteria, there have been cases up to 10 kilometers away from the source. That's the one area where we know. I think the brevetoxin, we were talking about it in the car coming over, how far away from the beach people are suffering from the symptoms, and I don't know the answer to that one, and I don't know that anyone's described that. I asked you, and you said it was it was even exhibited in the middle of Captiva and so forth. Mm-hmm. But does anyone know how far away from the water you can still get the symptoms?
I know people have tested as much as a mile inland, and they still saw brevetoxin in the aerosol. Mm-hmm. That's as far as people have tested, as far as I know. How much further it might be making it inland, I'm not sure. Okay.
So there probably is some more testing, some more research that needs to be done on that topic to get some more definitive ideas. Are there any treatment technologies for aerosolized toxins of either variety, brevetoxin or BMAA?
I don't think, I mean, we've talked about masks, and it's the Chinese all wear masks as far as I can see, but I don't think that this is going to happen to the American public. No, I think prevention is obviously the key here.
Just make sure Glade doesn't come out with the spirulina air scent. We'll make a buck on anything here. Can children get ALS?
Basically, no, although the youngest case that I've ever seen was 13 years old at the beginning of her disease. But this is still a disease of the elderly, or at least the mature. The incidence begins to rise around the age of 40, and it gradually increases at the age of 75. Alzheimer's, as you know, begins around the age of 50 and keeps on rising until you reach 105, unless you live in Ogimi.
Yeah. Eat all that seaweed. Are there any tests being done on seafood from that might have been affected by the Corexit and the Horizon oil spill, or from oil that still remains on the bottom of the Gulf? Does anyone know? It's not exactly a...
I'm not sure. I'm sure, I mean, so much money has been spent on that. I'm sure it has been, but I've not been following that research, actually. So I don't really know for sure.
Is there a viable technology, and I apologize if I'm not going to get this right, LG Sonic, an ultrasonic wave emitting machine that might destroy algal species?
There are actually commercial sonicators for lakes. They're usually in the shape of a swan for some bizarre reason. I don't know why. So this swan goes around the lake, sonicating the lake.
It's not as scary as a Martian.
Almost, yeah. But obviously the big problem with that is if you've got cyanobacterial cells in the lake and you sonicate them, you break them open. If they contain toxins, the toxins go into the water. Also, there are things like zooplankton that eat cyanobacteria. They also can get blown apart by sonication. It does exist. I've seen a few papers and studies looking at their use in lakes, but it's never really caught on as something that's very useful for controlling blooms. And as Larry said, these are old organisms. They're canny things. They'll survive a lot, and if you disrupt them briefly, at some point they'll come back, and probably come back harder as well.
And it wouldn't remove any of the nutrients from the water body, so those could
Those nutrients are still there, waiting to be used. And the other thing as well is with these ecosystems, if you've got cyanobacteria being eaten by zooplankton, you can also have fish that eat the cyanobacteria. You can have fish that eat the zooplankton. You have this whole beautiful system of control. And if you start disrupting it, then potentially you can shift it in the wrong balance, and suddenly the cyanobacteria have nothing to be competed against.
So what's it going to take to get laws in place to provide additional testing for toxins, or to set standards that would be more protective of water quality? We've talked about the nutrient impacts affecting both of these kinds of blooms. There seem to be a variety of sources. Are there any other countries, any other cultures that have effectively dealt with this? Are there any thoughts on the panel for what's really needed?
There are countries that have dealt with this. Florida's not unique in having cyanobacterial blooms. I want you to realize that it's a global problem. Many countries around the world have had this. As Rayanne mentioned, the WHO, the World Health Organization, has, and John mentioned it as well, has guideline values for drinking water for certain toxins. Microcystins are included in that. They have recreational guidelines. If you're not using it for drinking, but you can go swimming or fishing, then they have guidelines for that, either in the amount of cells that are present in the water or the amount of toxins that are present in the water. There are guidelines for microcystins for both drinking and recreation. There've been talk about guidelines for spermopsin, which is another toxin that I think, do you have spermopsin in Florida? Yeah? It's in the lake. So again, there's another cyanobacterial toxin. In the European Union, they have guidelines for things like saxitoxins in shellfish. They have guidelines for domoic acid, okadaic acid. All these things are there. It's a matter of what information do you need at what point in time. If you're using a lake for drinking water, then drinking water guidelines may make sense. If it's just being used for recreation, then you should perhaps think about putting in recreational guidelines. The WHO has no legal clout. It just gives you information to say, this is what we think based upon the best scientific evidence of what you should be working towards.
But let me take a broader picture of this, because Florida is really the epicenter at this particular time, and certainly from our interest point of view, with regard to these blooms. And we, I think, already know what is the major cause of these blooms, and it is the increase in the amount of nutrients which is getting into our systems. And the particular blooms that are occurring each year now on the east and the west coast of Florida come from the water which is coming out of Lake Okeechobee and is being rapidly shunted into the St. Lucie and the Calusa to be released and then to produce the nutrients that these organisms are blooming upon. We need, I think, a political process in this state which will begin to do what, in fact, was originally intended, which is to divert those waters not to the coasts, but to come down the Everglades and reestablish the Everglades. And that is a political problem in this state, because by and large, there has been a resistance for all sorts of regions to that answer. And that's why I'm delighted that Representative Fitzenhagen is here and is obviously going to I hope, sponsor the moves which are going to be following through what the public voted for, what was it now, 7 years ago?
2014?
14 years ago, to in fact stop these lateral flows and to stimulate the flows coming down south. So it's the politics that I think we are going to have to hope for is going to be preventative.
All right, since you took us there, let's jump right in. Questions about, in other places, industrial polluters are liable for damage they cause to the environment, public property, and putting people at risk. Clearly, this is a massive ecosystem that starts up in Orlando and comes through Lake Okeechobee to the Everglades and Florida Bay and has discharge points out the two estuaries, the Caloosahatchee and St. Lucie. The system has been designed to use those two estuaries as outlets for excess water, but we're in the process of trying to re-establish the needed projects to stem some of that harm. But the question is about responsibility and how some landowners are held harmless, while others are put at risk. And so, to be quite frank about it, the inland communities south of the lake versus the coastal communities on the two coasts. Is there a better way for us to balance those interests and share the water, share the responsibility for cleaning up the water?
John?
He started it.
Yeah, I guess I'm the natural in this one. Shared adversity is an issue that, if these events were occurring once a decade or once every 15 years, you'd say, yeah, we'll get through this, but they're happening all the time now. And so there are special interests that have perfect flood control and drought-proofed their crop systems. And so this concept of shared adversity is really a difficult one. It brings in some social issues, some science issues. It's very complex, but clearly we're not sharing adversity in Florida.
So following on that, we have a question: how can U.S. Sugar be a better partner in Everglades restoration?
I kind of feel like I'm being set up here.
You're not the only one with a microphone,
Yes.
but feel free to share.
Okay, I'll jump in real quick. There is some thinking now that the elevation of water in the EAA that would flood the towns and the level that it's currently at represents a storage condition that represents hundreds of thousands of acre-feet of water. And there's legal issues, judicial issues. I mean, it's very complex, but sometimes we call that dispersed water storage, and we don't call it flooding. And we pay farmers to store water on their land. Why can't we pay fair is fair? I have some other thoughts about that, but I think we could establish a situation where there is some reasonable compensation so that we're sharing these problems equitably, and it's just not happening now.
So one of the problems is that the inland farmers haven't been willing to sell their land. They've changed their position, and they're not willing to sell their land. So your proposal or this idea of water farming allows, if I'm understanding this correctly, allows people to continue to own the land, but we pay them instead of for all the costs of diverting water, we pay them to actually hold some of that water in the historic Everglades where the water used to go.
Absolutely. The concept is viable, it's equitable. I think it's a policy that our government could support. If you look at just the amount of economic damage that's happening on both coasts, it's so extraordinary that you have to look at alternatives that's going to bring some relief. And so, I get almost kind of emotional about this because I've been doing this for a very long time, and we use the word shared adversity an awful lot. But the concept of dispersed storage is a valid one. Those that have looked at it already, there's several 100,000 potential acre feet there, and the farmers are already flooding their fields 2 to 3 times a year for nematode control. So there is the infrastructure available to do it. We're just going to have the political will to dive deep into that situation a little bit more. We have representatives who do have political courage. One's here tonight, I think. But we need to go there sooner than later.
Sit at the table together?
I just wanted to, and I'm not a defender of big sugar, but the cattle industry, which is predominantly to the north of Okeechobee, is another contributor to the nutrient inflow. And both of those industries have to be addressed in this particular regard.
Yeah, improved pasture is the land use that has the single highest loading coefficient of phosphorus. So it's not just sugar cane, and it's in our upper basin as well as the EAA, to be fair. Yep.
And we do know that the land that is to the north of the lake has higher loading. Of course, there's a gravity pull in, but from those basins, that's been well documented through the years. And because that water is gravity fed into the lake, that's the only place it has to go. Whereas moving water out of the lake gives us a chance to reduce those estuary discharges. There are a number of projects that are out there getting started. We have a bill before Congress to to approve the EAA reservoir plan. How much will that help?
Well, the numbers I'm hearing are that somewhere around 65, 67% improvement from that reservoir. And I give the captains a lot of credit for really pushing that through the legislation. We have a similar reservoir in our basin that we've been a little bit critical of over the years in some contexts, but it will bring some relief. The thing that concerns me about these enormous above-ground reservoirs is when the agencies identify performance in terms of improvement, it's always in the context of everything else working in concert at the same time. When you ask yourself, how long is it going to take to bring all those other projects online? We're talking about 2-3 more decades. It is completely unacceptable for us to wait 30 more years to bring relief.
I'd like to add to that also. I mean, these reservoirs that are being built, they will deal with the water quantity during flood periods, reducing the flow. But they're reservoirs. I mean, I think of them as like little Lake Okeechobees. The nutrients are still there. If you want to get rid of the nutrients, you've got to have a marsh, such as the original Everglades, the original northern third of the Everglades.
So the current reservoir is approximately 240,000 acre feet of storage with about 6,500 acres of stormwater treatment marsh. We recognize that we could use more than that. This is a baby step going forward, but maybe this is where water farming gives us an opportunity for a win-win-win, because when water can be held and sit, it can drop nutrients, plants can absorb nutrients, sediments can settle out, and there could be opportunities for us to move water south out of the lake, get it moving slowly, treating the way Mother Nature once did, and allowing us to get water to the Everglades and Florida Bay that have been cut off. How are we doing on time? Keep going. Okay. Let's see. How can we make the general public more aware of the risks we face from these cyanobacteria blooms and red tides?
So let me address that one, because there is, I think, in our political organization, a concern that is a conflict, if you like, between public safety and the welfare of the state. And this is a thing that I think we need to address. This state lives by tourism, and if we give rise to public panic with regard to these blooms and so forth, it's obviously going to hurt the economy of the state. The representative knows this. On the other hand, if we don't look after the health of the people in this state, then we are not doing what the Department of Health and our legislature should be doing. And somewhere we have to achieve a balance. And as I said earlier, that in the end is politics. And somehow we're going to have to find that way to work this fair balance.
I'm picking up some of these. This one's out of order. I missed it before. How does the flying fox survive ingesting the cyanobacteria? How many species of cyanobacteria are there?
So, you answer the species.
Okay. In terms of the species, my guess, based upon what I've read, is there about 10,000. Does that sound about right?
Yeah.
But they grow everywhere. They grow on snow, they grow on deserts, they grow in plants, they grow on fresh waters, they grow on marine waters, they grow everywhere. So they've been around for 4 billion years, and there are quite a lot of them.
So I don't think we can therefore get rid of cyanobacteria entirely from any part of the ecosystem. The question is the amount of them, and that's what we're suffering here on the west coast from the bloom, and similarly on the east coast. What was the first part of that question?
The flying foxes,
Oh,
how
yes,
are they
sorry.
managing to
That-
survive that
That's a
cyanobacteria?
question that's been there ever since Paul Cox and his colleagues discovered this link with the flying foxes. And it is probable that there are ways that different animals and different insects and so forth have found to be able to live with these toxins and not be damaged by them. And they've developed enzyme systems and things like this to avoid the toxicity. What we need is humans to be able to develop those enzyme systems.
So should we all switch to sweet potatoes to get our Elserine? What are the other foods that we should be eating to maximize our exposure to Elserine?
There's, how do I put that? I live next to Idaho, so I can't
Oh,
criticize too many potatoes. No, I'm kidding. Sweet potatoes, I think, are fine. If you like fries, then you can eat more sweet potatoes. They certainly seem to be more higher in Elserine content from our analyses. From our studies of the people in Ogimi, and these were quite amazing people. Something like 8% of the women were over the age of 100, and they were all very fluid, as you saw in the movie, had very good recall, and we analyzed their diet. They had a lot of seaweeds that they collected themselves, which they've been collecting for many, many years. We also know as well that they were eating cycads during certain periods of time when they were starving, and so we know that they were exposed to BMA as well. They eat a lot of edamame, again high in serine. They eat a lot of tofu. They eat a lot of pork, so eat more bacon. Is maybe one way of going for it.
That bacon sweet potato
Yeah,
donut
bacon.
is
There
just
you go.
calling
Some
you.
edamame on top. So I mean, yeah, there are things that you can eat, some healthier than others. But I mean, I like sweet potato fries, and I do, if I feel inclined, I do choose them when I have my burger, like Dr. Cox said.
There you go. And you don't even have to fry them.
Exciting.
Does the federal government recognize the findings from the event in Desert Storm for the health impacts?
Yes, I think that the science and the actual figures did convince the federal government, and within about 5 to 8 years after the peak of Desert Storm, they set in place the VA ALS registry and made ALS a condition that the VA would provide all the services for and also provide disability payments for. So that I think has had a positive outcome.
So there are there other wars where there have been those clusters of illness?
Yes, I sat on an Institute of Medicine committee that reviewed all of this, and in fact there is a signal of increased ALS which goes right the way back to the First World War. It becomes more obvious further into the later wars, but there is something about being in the field or being in the military which does in fact increase the risk of developing ALS.
Wow. Can you discuss the term chronic and what chronic exposure is?
So I was telling Larry about this, a paper that is in the cancer field but is relevant to that question. There was a period in, I think it was in Denmark, where the water supply of a fairly large community was contaminated with arsenic that the people didn't know about until about 10 years into that contamination. And then they changed their water source, their water supply, and went to a water source which had no arsenic in it. So for 10 years, this community was exposed to arsenic at really high levels. They did not see until about 10 years after the termination of that exposure, they did not see an increase in cancer. But after about 10 years, the incidence of cancer went on rising for at least the next 20 years afterwards. So this is relevant to the question of how long do you have to be exposed and how much do you have to be exposed to, and when are you going to see that increase in the disease after you have been exposed. So in terms of diseases like Alzheimer's disease, Parkinson's disease, and ALS, we don't know how long that exposure has to occur before you start to get an increased risk. We don't know how long you are going to have to be away from that exposure before you're going to, in fact, stop having that increased risk. If you look at Guam, the story there suggests that perhaps 5 to 10 years is the period where you are at increased risk of, when you have exposure, of developing those diseases. But the evidence is very small, and there is a lot more research that needs to be done.
Great. One last question before I ask the panelists to give us their 2-3 takeaway messages. So the question is, can boats spread algae when they travel through it and take it into an area that doesn't have it?
Yes, and even birds can do it as well. There's reports saying bacteria being transferred on bird feathers. So, I mean, it's possible.
Okay. No one's immune. So now, in wrapping up, I so appreciate the ability to have these folks here that we can't hear from every single day, and to be able to have interaction. I would just like to ask you to give us some takeaway thoughts. 1, 2, 3 things that you'd like us to remember or focus on from your area of interest, or from your studies in this film and working in the environment, that you can share with the public.
Well, since I'm nearest to you, I'll start.
That's great.
I think what Paul Cox said in the documentary is true. Don't swim in the green, mucky algae. And the second thing that really is a corollary to that is that, in fact, one should try to move away from it because we now realize how far it can spread. And then the third thing is we need to find ways to be able to prevent them.
Thank you, Walter. For me, I mean, communication is essential. If you're going to deal with this problem, you've got to communicate, whether it's with people taking samples, people analyzing samples, people making decisions on what should be done with waters. And you, the general public, you're the people that also need to know what's happening. So that communication is essential. Secondly, panic is probably not the best thing to do. And thirdly, with the right analysis, the right guidelines, the right alert levels, where everyone knows what to do and when, you can safely deal with these risks and ultimately protect yourselves as best we can.
Like I said earlier, in one sense, the brevetoxin is a good one in that you know right away you're being exposed and you can now reduce your exposure. As I mentioned, BMAA is a bad one because you have no idea you're being exposed. I won't give the reasons here, but there is good reason to believe there's a bunch of other toxins in these cyanobacteria, which we have not discovered yet. I should point out, it was pretty much by accident we discovered the BMAA, and it's a brilliant work of Paul Cox that figured this out. It took many decades. Almost certainly there's other toxins in these cyanobacterial blooms we have not discovered yet that, again, do not have any immediate effects, so you don't know you've been exposed, but could have long-term health effects such as cancer, neurodegenerative disease, and so on. So I would say, first of all, I mean, the government agency is only measuring microcystin out of all these other potential toxins. Do not rely upon those tests and say, oh, well, if they don't find microcystin, it must be okay. For me, I think I would assume that there probably are toxins in those algal blooms. If you see a bloom of cyanobacteria, my view is that you should assume there are toxins in there. You don't want to go swimming in that water. You don't want to eat the seafood out of the water. And we don't have enough evidence yet, but I suspect you want to avoid breathing that air as well. The other point I'd like to make, again, once you've got the nutrients in your water bodies and you've got these blooms, there's not a whole lot you can do about it. You really need to stop them before they start. You've got to stop those nutrients before they get released from their source.
Well, we've had 2 incredible cyanobacteria events in the last 2.5 years that required the governor to issue a state of emergency in multiple counties. And we're currently experiencing perhaps the most toxic, perhaps the most pervasive red tide event, perhaps since at least 2006 and even earlier. So my takeaway is I really hope we can learn from these events, the impact that it's had on people that become so emotional and angry about these things. The 2016 bloom, we should have learned more. And I think after what we're currently experiencing now, I see it as an opportunity to really grow and meet some of these challenges. And I think there's some folks out there that can lead us in that direction. And I'm confident that we can meet these challenges in the future. It's going to take some political courage, it's going to take some imagination, it's going to take some high-powered science to do it, but I think we can. I believe we can.
Really appreciate our panel and all the wisdom that they brought to us tonight. I appreciate that all of you came out and have availed yourself of the information and becoming informed about these issues. Now we have a choice. We have an opportunity to observe the words of the Hopi elder, sort of as Paul Cox started this, I heard him say, you know, the native cultures had some wisdom that they relied on, and he wanted to build on that and find connections. In the same way the Hopi elders have said, there is no one coming to save us. We are the ones we've been waiting for. And so I just encourage us to all stay involved, stay engaged, be informed, and again, when conditions improve, don't go away. Don't go back to the old TV set and couch. Stay engaged. And again, thank you to Representative Fitzenhagen for bringing this great program tonight.